Hello, YouTube. My name is Miguel Atilano, and I attend Cal State University San Bernardino. Today, me and my partner, Jasmine Brinsky, will be giving a tutorial on how to do the Nanodrop procedure on cultured mouse embryonic stem cells, or MESCs for short. To start off in this procedure, you need to obtain your materials you will be needing, and the materials are MES media with no LIF. The reason there's no LIF in this nanodrop procedure is because we do not need to maintain pluripotency in nanos since the stem cells are not in contact with anything while hanging. The second material is PBS. The third, 15 ml conical tubes. 4, of course, you'll need your mouse embryonic stem cells. 5th, you'll need 2 petri dishes. Once you have all your materials obtained in arm's length of your hood, you will then want to sterilize your hood. And of course, we're going to do so with 70% ethanol and Kim wipes. And we will want to do this very thoroughly for no bacterial contaminations. Once you feel as if you have sterilized at the best of your ability, you'll next want to add all of your materials needed neatly in your hood. And remember to sterilize every piece of material needed prior to adding in the hood. Now that all of that is done, you are ready to start the nanodrop procedure. Start by opening up one of your 2 petri dishes and place the lid tilted on the bottom portion of said petri dish and add your mouse embryonic stem cells to the lid. The MESCs should pull up nicely on the lid. Now open your second petri dish. This dish is the one we will be placing 27 microliters of 60 to 80 drops of the MESCs onto it. Once all drops are placed onto the lid, you now need to add the PBS to the bottom portion of the petri dish. Make sure that the PBS covers all of the bottom of the petri dish. There should be no air pockets. Once all drops of the MESCs and the PBS have been added, you are ready to do the dreaded flip. When doing the flip, make sure that you're confident in your actions. Being confident helps out greatly. Being hesitant leaves room for error. So make sure to be confident and swift in your movements. I use the 1-2 flip method, and that has helped me greatly. When the petri dish is completely closed, you are ready to make your way towards the incubator. Be mindful and be careful of your surroundings as you make your way there. You do not want to lose any MESCs into the PBS. Once you get to the incubator, place the petri dish carefully inside and let it incubate for 2 days. After the 2 days have passed, you are now ready to do the harvesting portion of the procedure. You will need the following materials for harvesting: DMEM with 15% fetal bovine serum, FBS, 15 ml conical tube, and a tissue culture flask. Once all of the materials are in arm's reach of the hood, you will now need to start the sterilization process. And of course, we will be using 70% ethanol and Kim wipes, just as before. Once completely sterilized, make sure that you have a pipette aid and serological pipettes handy for this procedure. Once the hood is sterilized, we are now ready to start the harvesting process. We will first start by taking the lid up off of the petri dish and placing it gently on the bottom piece of said petri dish. We want to place it so that it's tilted. Once the lid is placed tilted, you now need to get the pipette aid and add 1.5 dmem with 15% FBS to the lid. This will add up to having approximately 3.5 ml of media on the lid of the dish. We are adding the DMEM to rinse the embryoid bodies off of the lid. Be patient while we're doing this, and make sure to rinse all of the embryoid bodies off. This usually takes about 10 minutes. Once you feel as if you have harvested all of the embryoid bodies and you don't see any tiny grains on the top of the petri lid, then you are ready to take up all of the media and place it inside of a conical vial. When the embryonics are inside of the conical vial, we need to make sure that we leave 3 to 5 minutes for the embryonics to settle. Once the time has passed, you are now ready to add the media into the tissue culture flask. Now that the embryoid bodies are successfully inside of a tissue culture flask, you are now ready to take it to the incubator. Place it carefully in the incubator and allow to incubate for further research on the cells. And you have now successfully done the Nanodrop procedure, and you have successfully cultured embryoid bodies. Thank you guys for sticking around to the end. If you have any questions for me and Jasmine, we'll gladly answer them in the comment section below. I'd like to give a special thank you to Dr. Bornius and Dave Coffey for offering tissue culture at Cal State University San Bernardino. It's been such a great experience. Thank you guys both.